gastrointestinal tract is a vital component of the body's innate immune system. Epithelial and immune cells monitor the microbial environment and decide how to respond to different types of bacteria. Differential recognition of bacteria is made possible by cell surface proteins called pattern recognition receptors, or PRRs. These receptors detect surface motifs that are present on some bacteria, but not others. A good fit tells the receptor to activate the production of cytokines, chemokines, and other immune mediators. Toll-like receptors are an important family of pattern recognition receptors. Toll-like receptor 4 recognizes a pattern called lipopolysaccharide, or LPS, which is found on gram-negative bacteria. A single nucleotide polymorphism in the TLR4 gene reduces the receptor's ability to activate an immune response in the presence of LPS. The clinical implications of this variant remain inconclusive. Some evidence suggests a possible effect on microbial composition in the GI tract. In clinical settings, this phenotype can be confirmed using common microbial assessment methods such as the urea breath test or stool PCR testing. On the other hand, the variant may have a desirable effect on cells and tissues because it reduces cytokine production. More research is needed to clarify these phenotypic associations and their clinical translation.